I'm breaking it down into pretty small pieces, right? We'll see at the end there, putting it all together, how you want to keep this kind of flow of information, even if I didn't do things quite in sequence as we went along, right? Because we started with the anatomy of the sarcomere, right, which included that Z disc where the actin filaments were attached to, and then the actin filaments interacted with the myosin filaments over here, and they interacted with them and pulled them over toward this M line, right, along with the Z disc, right? That was the actual contraction of the muscle, right? And that interaction between your myosin head and your actin filament was a calcium dependent reaction, right? That calcium was going to be coming from your sarcoplasmic reticulum in response to a electrical signal coming down the T tubule, right? And so these names here, if you're looking at any other book, if you have another physiology, you have other references, you might see these, but this is just the voltage gated calcium channel, the DHP, dihydropyridine, L-type calcium channel, but don't worry, I'm just calling it a voltage gated calcium channel. And then the ryanodine receptor channel over here, which is your calcium release channel on the sarcoplasmic reticulum. Again, I'm not asking you these. Your book doesn't have these names. Voltage gated calcium channel on the T tubule, and then the calcium release channel on the sarcoplasmic reticulum. And upon the electrical signal coming down from the action potential, there was some kind of conformational change where this was open, calcium spilled out, and that changed the interaction between your myosin and actin filaments, right, through these regulatory elements. And we saw that action potential traveling down the T tubules from the sarcolemma, right? Today, we're going to look at this whole interaction a little bit over here, how this signal starts. All right, and then at the end of this, we'll also talk a little bit more about how the muscle generates contractile force based on the length of the sarcomere. And so we have a nerve coming in here, right? That's attached to a certain region on a muscle fiber, right? This is the multinucleated striated muscle fiber, such that when it sends a signal, it's going to send an electric signal. It's going to change into a chemical signal and back to an electrical signal, which is going to go down the length of the muscle fiber pretty much simultaneously. All right, so we're going to be looking deep into this little area over here. So first, we want to, kind of remind ourselves from way back when about signaling, right? And about trying to reach target cells. And we learned about the endocrine system and hormones that were traveling through the circulating blood. And those hormones leaked out of the capillaries into the interstitial fluid. And when there was the cells that had a receptor for that hormone, right, that cell could respond to that hormone, right? So you had to have the particular receptor. The hormone might be going everywhere in the body. Its target cells would be the ones that had that particular receptor. Okay, but so it was a very general, widespread effect that could reach anywhere in the body, right? Just by spitting out some hormones from the pituitary gland, right? You're going to have these big, widespread effects, or from the adrenal medulla. Right? There's going to be circulating hormones. It'll find its target cell because that target cell will have a particular receptor for that hormone and not this one. So it'll respond to this hormone, not that one. General widespread effects, and you had this interaction. What was the effect of this binding? Well, this receptor, depending on what this receptor did, right? Maybe it led to this whole sequence of events, which led to changed gene expression or changed enzyme activity, or maybe insertion of glucose transporters into the membrane, something like that, right? That was the effect, right? This is the effector cell of the hormone. So that was hormones, right? General widespread effect. With the nervous system in particular, right, you had those specific wires coming out of the spinal cord and brain, right? And those wires went out into the body to very, very specific locations, and they targeted only the cells they were connected to, right? They were very closely connected to, so that this purple one didn't affect any of these ones that were innervated by the red one. The red one didn't affect that, right? They were very specific in the target. And as soon as this signal, there was going to be a signal, right? A little chemical signal over here. As soon as this neuron stopped that signal, it was over, right? Very, very, very specific and very brief change, right? In what on the effector cell. And that's what we're looking about with the nervous system. And this in particular, because we're talking about skeletal muscle, we'll see this is so-called somatic nervous system. Right. So these neurons, these axons came out of the spinal cord, out the spinal nerves. They branched out to innervate a couple of different specific targets. And those branches branched off into individual muscle fibers and ended, split off into this branch called the telodendria. Don't worry about this word so much. We'll get to this when we get to the nervous system. But they branched off and they ended at a very particular region on the muscle fiber. All right, so let's look at the muscle tissue in general, right? This is a cross section of, let's say, your biceps brachii. You're cutting in half, and what do you see inside there? You see the fascicles. And then you see all that connective tissue in between them. In there you have blood vessels, and you also have axons that have penetrated through the muscle. These are the nerve fibers running through the muscle and running down the length of the muscle. Right? And so they're running in through the muscle, and then each one of these is a fascicle, which is a bundle of muscle fibers or muscle individual cells. Right? So if you take one of these right here, here's where we get to the actual muscle fiber that we've been talking about, the muscle cell. This is that whole general organization, right? Tube within a tube within a tube, right? But what we're looking at here are the innervation, right? The axons running through it. Remember I said the connective tissue serves as a route for blood vessels and nerve fibers to run through. So that muscle cell was surrounded by an endomysium, that connective tissue covering, loose connective tissue. And then running through that endomysium, you had that axon, right? So in this case, here's the axon, but it would be running through the muscle, running down the fascicle, and then running and stopping somewhere over here, right? To get over here. That would be the axon of the motor neuron. Right? So here's a close-up of that. There's the telodendria, that is that final branch of the nerve fiber, and it ends on a particular region of the muscle cell called the motor end plate, right? So the motor end plate is that region on the muscle fiber. Right, so that telodendria branched off until it became, at the very axon terminal, the synaptic knob or synaptic boutons or synaptic terminal, any one of those. Looking at this cartoon picture from your book, right? This is the nerve fiber coming down over here. It was branching off into a couple of different little regions, but it was still confined to this yellow area. Right? This will be your motor end plate, and where these nerve fiber in the muscle cell meet at the motor end plate is the neuromuscular junction, a junction where two places meet. Right? So that is this over here, right? The neuromuscular junction. Here's the neuron, here's the muscle fiber. This is the junction where it meets, right? And so we're going to look very closely at that junction and see what goes on in between the neuron, what it does, and what the muscle cell does. All right. So just for the recap purposes, right, we talked about this last time, but it's worth to redo because, even though we're not getting too deep into it, it's useful to know because it'll be worthwhile when we get to the nervous system to really understand this whole concept of resting membrane potential and, and then the action potential, right? So remember, the resting membrane potential meant the difference in between the outside extracellular fluid and the inside intracellular or cytosolic fluid. There was a difference in the distribution of proteins and ions and stuff. And what we're interested in here is the charge. That is, there was more positively charged ions and stuff outside the cell than there was inside the cell. Right? This is largely due to maybe two things here, right? You had a lot of proteins and DNA that's kind of negatively charged. You had phosphate groups as negatively charged. And then sodium is basically kept out, positively charged sodium is basically kept out of the cell by this selective permeability, right? And the sodium potassium pumps that was constantly pumping out sodium. And by doing so, you built up this positive charge on the outside of it. So sodium would love to get in here down its electrochemical gradient and get to all this nice negative charge, but it can't unless given the permission to do so, right? So any cell that you happen to look at might have this kind of potential, right? But really, when we talk about nerves and muscle cells, we have this, we'll come across this number, minus 70 millivolts. That is, it's minus 70 on the inside compared to the outside, right? This would be like if this was the inside of a freezer over here and it was minus 70, the outside, if you outside of the room was relatively zero, let's say, and then this was minus 70. So that's the first starting thing you need for this excitability, right? And what we saw was that the excitable membrane, right? That's true for any of that membrane potential, could be true for any cell, but with nerves and muscle fibers, you had what's called an excitable membrane, right? You could excite them, you could pass an electrical signal down the length of the cell. So in that case, that excitability was due to the distribution of voltage-gated ion channels embedded within the membrane down the length of the cell, right? Or distributed in a very particular way down the cell, which meant that if there was any kind of change in the membrane potential, if all of a sudden, there was a influx of sodium ion, or in this case, potassium ion, now let's just say any voltage change right here, right? That would open up this gate, right? Because it would sense that change from minus 70 to minus 50. That was the threshold. The channel would open up. And in this case, it's a potassium channel. So it was permeable to potassium, and potassium would flow down its electrochemical gradient, which happens to be out of the cell, right? Because potassium is kept high relative to the outside. So it would flow down its electrochemical gradient, which is leaving the cell. This is the example of a potassium channel. What would happen if it was voltage-gated sodium channel? I just told you sodium was very high on the outside, low on the inside. If this was a voltage-gated sodium channel, all the sodium would flow inside the cell. If it was a voltage-gated calcium channel, calcium is also kept very low intracellularly. So calcium would also flow in here. So in this case, potassium's flowing out, but if it was a sodium channel, right, what's going to happen? There's a change in the potential somehow. There's a voltage sensing domain of that channel that is going to open up that sodium channel or ion channel, and sodium is going to go down its membrane, right? And the way it's going to go is that these are lined up in a series such that at the start, right, this depolarization event, that is like we just saw mine going from minus 70 to minus 50 millivolts, right? You're getting closer to zero. That's called depolarization. That signal, because of these ion channels, is going to spread out, right? In the case of a muscle fiber, it's going to spread out in both directions due to these ion channels here, right? Because you're going to get a small change in electric over here. That's going to open up here. That's going to cause a change over here, which is going to cause this to open up, which is going to cause a change and cause this to open up, which is going to cause a change and cause this to open up, right? And that's how you get this spread of voltage flips going down the length of the muscle. This is what we talked about last time, but we're just recapping it. We'll cover this in a lot more detail when we get to the nervous system. Always nice to learn things twice, though. It just cements it in. All right. So next question is, I talked about a depolarization event. Now we want to know where this starts from. Where does this initial depolarization event before that chain of voltage flipped occurs? Right? And so this is where we get to your neuromuscular junction, right? Because we're going to find these ligand-gated ion channels at the neuromuscular junction that are going to respond to a signal and then open up, right? And then outside, that's going to cause an influx of ions to come in there and cause that initial depolarization event. So that gets into these ligand-gated ion channels, right? They open up in response to binding of a ligand, right? Any kind of signal. This could be, theoretically a hormone. In this case, it's the neurotransmitter acetylcholine, right, at the neuromuscular junction. So in the next video, we'll look at how that electrical signal in the motor neuron is going to be converted to a chemical signal and then reconverted back into an electrical signal on the muscle fiber. All right, see you next time.